Hello, everyone. Welcome to today's webinar brought to you by Microbial Insights. This webinar is on assessment of MNA as a remedy for toxins released in the East Palestine, Ohio train derailment accident. We appreciate that you are taking the time today to join us, and it is my pleasure to introduce our esteemed speakers, Dr. Frank Loeffler and Dr. Gaio Chen. Dr. Frank Loeffler is a professor in the Department of Civil Engineering and Environmental Engineering at the University of Tennessee, Knoxville, and holds the Goodrich Chair of Excellence in Civil Engineering. He is an environmental microbiologist, and his group conducts fundamental research to better understand, control, and manipulate microbiomes involved in carbon cycling, nitrogen turnover, and contaminant degradation in natural and engineered systems. Dr. Gaio Chen is a Research Assistant Professor in the Department of Civil and Environmental Engineering at the University of Tennessee, Knoxville. Dr. Chen is an environmental microbiologist, and his research explores the physiology and ecology of anaerobic microorganisms involved in the degradation of chlorinated solvents and petroleum hydrocarbons. By applying integrated physiological, biochemical, genomic, and proteomic approaches, he identified novel microorganisms and pathways for bacterial metabolism of chlorinated methanes and ethanes under anoxic conditions, and he also elucidated how sulfate-reducing bacteria functionalizes and degrades aromatic hydrocarbons such as benzenes and naphthalene. Now, before I let Frank and Gaio take it away, I would like to remind you all, as always, this webinar is being recorded and will be available in the resource section on Microbial Insights website by the end of this week. So don't worry, we'll also be sending out an email with a link and a certificate of attendance following this webinar to those that are registered. So after Frank and Gaio are done, there will also be time for questions. So feel free to type your questions as the presentation goes on, and either I or Dora will try to read off as many as we can, so when that time comes, so they can get you their answers. So join us in welcoming true advocates for sustainable solutions, Dr. Frank Loeffler and Dr. Gaio Chen.
So thank you very much for this introduction. So I jump right into it. So we are going to talk about the possibility for natural attenuation as a remedy for these toxins released in the East Palestine, Ohio train derailment accident. So this work is a collaborative effort between my lab and Microbial Insights. And this work is supported by an NSF grant under the Rapid Response Research RAPID initiative. So here's the team, briefly introduced to you, Dora and Sam from Microbial Insights. Then we have Ana Arosemena and Brian Rosado Maldonado, two undergraduate students who helped last summer. Then we have Elizabeth Padilla Crespo, who is a professor at the Inter-American University of Puerto Rico, Agudila. She was here last summer and also participated. And then myself and Gaio Chen took the picture, so he's not in it. And then I would like to point out Paula Coelho and Andy Welton from Purdue University. So Andy is the professor, Paula is the student, and they also worked in East Palestine and were very helpful connecting us with local residents. That was a requirement for us to get samples. So on February 3rd, 2023, a 150-car train derailed near the village of East Palestine, Ohio. And 20 of the 53 derailed cars carried hazardous materials, including 5 cars with vinyl chloride and 1 car with butyl acrylate. So here is a list of the chemicals or products that were on these derailed cars. So you see vinyl chloride, 5 cars, about 1 million pounds, butyl acrylate, different glycols, and a few other chemicals, and also products like PVC, polyvinyl chloride, and polyethylene were on hoppers on these derailed cars. So 3 days after the accident, the decision was made to puncture high-risk cars and drain about 1 million pounds of vinyl chloride and 250,000 pounds of butyl acrylate into a makeshift pit for a controlled burn. And this burn unleashed an extensive smoke plume loaded with particulates, what is visible in this image, which penetrated the cloud cover and was in fact visible from space. Here taken from an airplane window, you see this extensive smoke plume. And I come back to that at the end of the presentation. So of these chemicals released, obviously of greatest concern was vinyl chloride, but also butyl acrylate. The other chemicals obviously are also of concern, but many of them are known to be readily degraded or not being recalcitrant in following release. So we focused on these two chemicals, vinyl chloride and butyl acrylate. And we assessed if the resident microbiota in this area where the accident occurred could degrade and detoxify these compounds. So just a brief background information about vinyl chloride. It's a gas at room temperature, but in these tanks, under pressure, it's a liquid, and it's relatively soluble in water, about 2.7 grams per liter. The image to the right I chose to show here just to emphasize that it's a poison, and this is an artwork that is displayed in the Andy Warhol Museum in Pittsburgh, Pennsylvania. Also to give you an idea where East Palestine is located, it's about an hour northeast of Pittsburgh, Pennsylvania. So health concerns associated with VC, we have acute effects in response to high concentrations, irritation of eyes, dizziness, drowsiness, headaches. It has an impact on reproductive and development effects. It has chronic non-cancer effects, and most important probably, it's classified as a human carcinogen. Bottom line, vinyl chloride is dangerous, it's toxic, and exposure to elevated concentrations and long-term exposure should be avoided. Now, why do we have vinyl chloride on trains anyway? So that has to do with the use of the VC monomer. It's predominantly used to generate polyvinyl chloride, PVC, in a polymerization process. And PVC is here, you see it, the numbers here are in millions of tons. About 40 million tons of PVC are produced globally every year. So it's a very important plastic. And it's mostly used in construction, PVC pipes, windows, and so on. And so now one important thing to realize is that the production of the VC monomer and the production of PVC are not co-localized. They happen in different companies, in different locations, in different states, meaning that that vinyl chloride has to be transported, mostly by train. And when you run a train in the US, it's highly likely that you sooner or later get involved in an accident. So I believe the numbers for 2022 are, we nearly had in the US alone, nearly 1,200 train derailments. So meaning about 3 derailments per day on average. So the likelihood of accidents is fairly high. And this train derailment in East Palestine, Ohio, is not unprecedented. There were similar accidents in 1982 and 2012 in the US and also in other countries where vinyl chloride tank cars were damaged and vinyl chloride released. So East Palestine is not a unique event. It has happened before. And so this map here gives you a brief or a rough overview of the area. You see here the derailment site. It's close to the border between Ohio and Pennsylvania and directly at a stream called a connector stream that discharges into Sulphur Run, a small stream that discharges into Leslie Run. Leslie Run goes south, runs into Little Beaver Creek, which ultimately discharges into the Ohio River. And so now the question for us was, can the microbiomes in these streambed sediments degrade the contaminants of greatest concern, namely vinyl chloride and butyl acrylate? And so we basically explored the question, is natural attenuation contributing to contaminant degradation and detoxification? So natural attenuation relies on natural attenuation processes within the context of a monitored site cleanup strategy that will reduce contaminant concentrations to levels that are protective of human health and the environment within a reasonable time frame. That's a definition slightly modified, originally released by the EPA. So now these natural attenuation processes can be physical processes, chemical processes, and biological processes. The focus of our work was on biological processes, specifically microbial processes that can act on these contaminants. And so with the RAPID grant that we were awarded, so we received the funding in early July. So we went in June and November on 2 sampling campaigns to East Palestine. And here are just a few images showing the team collecting samples. There are 2 things I would like to point out on these images. First, this here shows this connector stream, and you see there's very little water. A day later, when it rained at night, to the right here, this is the connector stream after a rain event. So you can see that the conditions here change dramatically depending on water level, rain events. It also means for us taking samples, it's most likely very heterogeneous where we sample because the conditions are changing dramatically depending on rain events and weather. I also would like to point out this image here, where a local resident shares his experiences with the accident, the contamination that they observed in the creek. And so in discussions with local residents, we learned a lot what can help us understand natural attenuation in this watershed. So now I turn it over to Dr. Gao Chen, who will be talking about the sampling strategy and the setup for testing, for applying molecular biological tools and testing, performing a treatability study in the laboratory.
And so following the accident, the EPA conducted a surface water monitoring program. Here's a map showing, their monitoring locations. So the blue dots showing their background monitoring location, and the green dots show the monitoring locations. Here, the red dots show you the derailment site. So they have several monitoring site locations just around the derailment site. And based on the EPA monitoring program, they can observe that, in the downstream of the accident, these monitoring sites, they detect high concentrations of VC, is reaching around 8,000 ppb in some locations. And the high concentration of VC only occurred on the downstream, like the sulfur run and also in the Leslie run. And the VC concentration decreased quickly, after 2 or 3 weeks, and decreased to very low concentration or non-detectable concentration. And so here I just show that the EPA did detect VC in the surface water. And VC can be degraded by microorganisms in the environment under oxy conditions with the presence of oxygen. It's known that several microorganisms can degrade VC through metabolic and co-metabolic process. Metabolic process means the degradation of VC is coupled to the growth of the microorganism, and a co-metabolic process means the degradation of VC relies on a primer substrate like a methane, ethene, or ethane. So there are several types of bacteria has been identified can degrade VC and oxy conditions, such as mycobacteria and myconicolbacterium and Nocarditis, rhodocos, and some others. And the degradation pathway has already been elucidated. And first, the VC is converted by a molloxygenesis to an epoxide. And the epoxide can be converted to a coam adduct. This reaction is catalyzed by a coam transfers. And these two steps are the key steps in the VC degradation pathway. And they are encoded, the enzyme are encoded by ETNC and ETNE genes. And these coam adducts can be further degraded to acetyl-CoA and channeled to a central metabolic pathway. So this is a degradation pathway under oxy conditions. Under anoxic conditions without oxygen, VC can also be degraded by a reductive dechlorination. So some bacteria, some dihalococoids, McCarthy strains, and also some dihalogenomolars can use VC as a terminal electroreceptor and reduce it to a non-toxic ethene. So this is called a reductive dechlorination, and the key enzymes has been identified. VC dechlorinase has been identified, such as TCEA, VCRA, BVC, and CERA genes. These genes are encoded VC reductive dehalogenesis. And these key genes like ETNC, ETNE, and this VC reductive dehalogenase, and also the 16SI genes of the dihalococoids and dihalogenomolars has already been identified, can be used as monitor biomarker genes to probe the VC degradation process in the environment. So here we connect samples from connect sediment sample, surface water, and drinking water wells from these locations highlighted in this map. And so these reds are the sampling locations, these symbols here. And we did a sample for 2 sampling campaigns in June and 1 in November. So our first sampling campaign happened in June, which is already 4 months after the accident. So after we took the samples, we performed molecular biological tools. We extract DNA and we measured and monitored the biomarker genes using unquantified area high throughput qPCR assays. And this work was done by Microbial Insight. And we also did microcosm treatability studies. We we focused on VC and butyric and try to see if the native microbiome in these samples can degrade these 2 chemicals. And here are some results of the qPCR detection of VC degradation biomarker genes using the targeted qPCR assay. So in total, we connected 22 sediments and 22 surface water from this area, and we detect the dihalococoids in 64% of the samples, and with abundance reaching 10 to the 6 copies per gram in a sediment. We also detect the dihalogenomolars in 55% of the samples, reaching the abundance reaching 10 to the 7th copies per sediment. We also detect dihalococoids and dihalogenomolars in surface water, but the abundance are very low, and only a few samples were detected. And here, the plus symbol shows the detection of dihalococoids in these locations. and the star indicates, the detection of dihalogenomolars in these locations. We can see that, dihalococoids and the dihalogenomolars were not frequently detected in the samples and connected from the sulfur round, suggesting, these samples are exposed or adapted to the VC spill, and this group of microorganisms probably already been enriched. And one thing surprising, we did not detect VC RDSs or detect in a few samples, but abundance is below detection, below limit of quantification. So for the ETNC genes, the detection frequency is much lower than the dihalococoids and dihalogenomolars. For ETNC, we only detect like 20% of the samples, and ETNE detect in like 15% of the samples. The abundance reaches 10/5 copies per gram sediment. We did not detect these two genes in the water samples. And here is shows that where these genes were detected, it's the same as dihalococoids and dihalogenomolars. They were frequently detected in samples from sulfur round, and or suggesting, these microorganisms have been reached in this environment. And yeah, we're surprising finding that we detected the dihalococoids in all well water samples, the drinking wells water samples we connected from a resident property. So we monitored 5 drinking wells. Like here, these are the drinking wells. So we have 5 drinking wells. We connected at the 2 June and also November. One of the samples was not connected in November. We detected the dihalococoids in all of the samples except one is below limit of quantification. And the abundance of dihalococoids is low. It's around 0.3 to around 5.6 cells per mil. And here, this gray bar is a sulfur reducing bacteria. So we also detect quite high numbers of sulfur reducing bacteria in these drinking wells, reaching like 10 to the 4th. And the blue bar shows the abundance of total bacteria. So it can reach to like 10 to the 7th cells per mil. So what it means for the detection of dihalococoids in the drinking well waters, we know that vinyl chloride was never detected in the well water, and dihalococoids are strict organohalide respiring bacteria, means they can only live with chronic solvents as a terminal electron acceptor. So it is unlikely they live or grow in these drinking wells. And we hypothesize that maybe there's a connection between the surface water and the groundwater through some fractions. There are not of fractures maybe in the rocks aquifers, as shown in these red lines. And when the accident happened, the VC spilled, and there's a bloom of dihalococoids. Dihalococoids bacteria might grow in biofilm, and these dihalococoids might migrate and transport with the water flow, and then get into the drinking well, then stay there. And that's the reason why I detect this group of bacteria in the drinking wells. And also by talking with the residents, we found residents showed us that they report they can smell a rotten egg smell in their drinking well, suggesting there are sulfide present in these drinking wells, and it agrees with the detection of high abundance of sulfur-reducing bacteria in these wells. So it also shows that there's a connection between the surface water and the groundwater. After the accident, so some other chemicals spilled, and they can migrate to the groundwater and serve as an electron donor to stimulate the growth of sulfur-reducing bacteria, generating sulfide, so which gives them this rotting egg smell. And the residents said that this smell did not occur before the accident. So, and then yes, we performed microcosm treatability study, and we focused on first vinyl chloride. We test the vinyl chloride under oxic and noxic conditions, anoxic conditions. So we have oxygen in the H space serve as an electron acceptor, and VC was added as an electron donor. We add sediment as a source of microorganisms, and the microcosm was definitely with an autoclaved mineral salt medium. And anoxic condition, we add lactate as an electron donor, and VC was added as a electron acceptor. So the sediment was connected from all these locations shown in the map. And we found that anaerobic VC occurred in all sediment microcosms. You can see here, VC was degraded in all microcosms. And here, these are the autoclaved controls. If we this microcosm set up with autoclaved sediments, we don't see a complete degradation of VC, just neither a loss. And the VC was degraded after a neck phase of around 20 or 40 days, and it was completely degraded in the microcosms. And after degradation, we spike a new VC, fresh VC, and the respike VC was quickened and degraded in these microcosms. So, this study shows that anaerobic VC degradation or aerobic VC degradation is widespread in all these microcosms. So, and then we performed 16S RNA gene amplicon sequence to identify which group of organisms are likely or responsible for aerobic VC degradation. We detect in several or many oxic VC degrading microcosms. We identified mycobacteria. In some microcosms, they are pretty dominant, reaching the abundance of 35%. So in a total amplicon. So, and in some microcosms, we detect the mycobacteria, and the abundance is low, it's 0.2 to 0.6%. We also detect some other group of microorganisms, which also known as VC and degrading bacteria. So they probably they also play a role in VC degradation. So mycobacteria is frequently detected in or frequently isolated in oxic VC cultures. So it serves as a model organism for oxic aerobic VC degradation. So then we perform the qPCR for these oxic VC degrading microcosms. So in the sediment, we don't detect ETNs, or these bars represent ETNC and the abundance of ETNC and ETN genes. In the original sediment, we used to set up microcosms. We only detect ETNC and ETNE genes in very few sediments, like the sediment from location 2 and the sediment from location 4. Both of them are from sulfur, and so in other sediment we don't detect these genes. But after the enrichment in the microcosms, these ETNC and ETNE genes were detected in high abundance, suggesting that during the enrichment with VC, we are enriched microorganisms carrying the ETNC and ETNE genes, and suggesting their involvement in aerobic VC degradation, and also suggest that ETNC and ETNC can be used as a biomarker for aerobic VC degradation. And also we started VC degradation in anoxic microcosms. So we performed microcosms without oxygen, and we add lactate as an electron donor. So the reductive decoordination of VC to ethene was readily recovered in microcosms with sediment from location 2, 4, and 8. So you can see in these microcosms, VC was degraded and the ethene was generated. So suggesting the reductive decoordination of VC is happening. And in microcosms with sediments from other locations, after several months incubation, we haven't seen activity yet. And then we performed 16S RNA gene amplicon sequence In the original sediment, we did not see dihalocolloids or dihalogenomolars in the sediment from locations of 2, 4, and 8. But in the microcosms, we detect dihalocolloids in abundance like 0.2, 0.5, and 0.3%. And in the first transfer cultures, we see the enriched dihalocolloids in these microcosms reaching like 5 or 6%, so suggesting dihalocolloids group were enriched in anoxic VC degrading microcosms. So dihalogenomolars was also detected in some locations, but in the microcosms, they were largely undetected in these microcosms, suggesting our enrichment process might introduce a bias. So in selecting dihalocolloids. And we also performed a target qPCR. We monitor the 16Si genes of these dechronators and also VC-RDS genes. And this is in the sediment, original sediment, we did not detect dihalocolloids. We detect dihalogenomolars in 2 of them. And after enrichment in the microcosms and also in the first transfer cultures, we see a high abundance of dihalocolloids and also high abundance of these VC reductive dehalogenesis genes. And so in agreement with previous amplicon sequence data, we see the decrease of dihalogenomolars or either non-detection of dihalogenomolars, suggesting the unfavorable selection of dihalogenomolars in this enrichment process. And butyracquinate is another chemical EPA monitored, and they detect a high abundance of butyracquinate. Like in these monitoring wells, it's similar as before, because the concentration is much higher than VC, can reach to 180 ppm. Oh, yeah. So and the butyracquinate can travel further downstream to even to the Ohio River. They can detect like a 500 in this location, detect like 470 ppb butyracquinate. So butyracquinate did occurred in this or spilled in that after the accident and occurred in this environment. So we did a microtreatability study for butyracquinate because butyracquinate is known very easily readily degradable on oxic conditions. So we focus on the anoxic conditions. So we added the butyracquinate as an electron donor. We didn't add any electron receptor in these microcosms. And we monitor the butyracquinate with GCFID. You can see that butyracquinate is readily degraded in all of our microcosms. So this square solid line shows the butyracquinate, and we also observed or detected the butyral. So the triangle dashed line here, you see they are transiently produced. So, and in autoclave control, there's slight disappear of butyracquinate, but there a little bit increase of butanol, but the degradation only occurred in the active microcosms. So here we show that butyracquinate can be degraded in the microcosms, and this ester bond can be hydrogenized, generate butanol. And in the second transfer cultures, butyracquinate is quickly degraded. You can see the origin line. So it's degraded within a few days. And in addition to butanol, the blue line, we also detect the acrinate, as a transient intermediate. And demonstrate, after the hydrolyze of the butyl acrinate ester bond, it will generate the acrinate and butanol as an intermediate. So we know that a group of esterases or hydrogenases can hydrolyze the ester bond of butyl acrinate, generate acrinate and butanol. This esterase or hydrogenase is not dependent on sensitive to oxygen. So these enzyme esterase or hydrogenase can function under anoxic conditions, and we know that this group enzymes usually have a broad substrate range. So so these acrylated and n-butanol can be readily completely oxidized or mineralized to CO2, and the electrons generated can use to reduce very common like electron acceptors. So this process can happen under different redox conditions. Without in our microorganism, there's no alternative electron acceptor added. So the acrylate and butanate can still be fermented. There are studies showing that acrylate can be fermented to lactate and then to acid and propionate. And these alcohols and fatty acid can be degraded under centrifugal oxidation, generating methane or some other non-toxic compounds. So so overall, so it just says, butyric can be readily degraded under both oxic and anoxic conditions. Here I will give back to Frank.
Okay, so Gao has demonstrated with this treatability study that these chemicals, vinyl chloride and butyl acrylate, can be readily degraded under oxic and anoxic conditions to non-toxic products. So demonstrating strong attenuation potential for these chemicals in the East Palestine watershed. For the example of vinyl chloride, he also clearly demonstrated the power of molecular biological tools, specifically qPCR, when targets are available. So knowledge about the genes that are involved in degradation or the enzymes encoded by these genes involved in degradation, then we can apply MBTs. But for the degradation of butyl acrylate, we don't know the specific genes involved in butyl acrylate degradation. So here we couldn't use MBTs, qPCR specifically, because the target genes are not known. And I come back to this in a second. So now I want to talk about something I mentioned in the beginning of the talk, and it has to do with the controlled burn. So whenever you burn something that contains chlorine, like vinyl chloride, there's a potential that you generate something very undesirable, like polychlorinated dibenzodioxins and polychlorinated dibenzofuranes. So for example, these chemicals are also generated when you burn polyvinyl chloride. And remember, on 4 of these train cars was polyvinyl chloride that was also burned. In addition, the burning of vinyl chloride, the monomer, can generate PCDDs and PCDFs. And so these chemicals, PCDDs, PCDFs, are also generated in forest fires. There's geologic formation. Even biological processes can contribute to the formation of these chemicals. But most likely, combustion processes are a major source. For example, unintentional formation in incinerators, also in chemical synthesis. And they're also introduced through accidents. So 1976, some of us may have heard about this. There was a catastrophic accident. A production plant for chlorophenols exploded. A reactor exploded in the small town of Seveso. Several people were killed because of the formation of these very toxic polychlorinated dibenzodioxins and polychlorinated dibenzofuranes. So also in the derailment accident, obviously, you have a fire, you burn polyvinyl chloride, can contribute to the formation of these chemicals. It's an accidental release. So now I call this here intentional. And this may be a little controversial, and the situation on the ground may have required that vinyl chloride has to be burned on site in a controlled burn. I call this intentional because it was known that there's a strong potential for the formation of these chemicals. PCDDs, PCDFs are toxins. They're endocrine disruptors. Some of them are known human carcinogens, very strong known human carcinogens. They are far more recalcitrant than vinyl chloride, and they bioaccumulate and have a tendency for biomagnification along the food chain. So I think the point I want to make is, if you think about what Gao Chen presented, vinyl chloride is a chemical that's toxic but short-lived. It can be degraded, for example, by microbes. It is also susceptible to oxidation in the atmosphere, so it's a short-lived contaminant. What we generate here, these are long-lived contaminants. They are recalcitrant. We have to deal with them for a much longer period of time. And I think that consideration should have received or should receive more attention. You know, we deal with something toxic that is short-lived versus something that is toxic and long-lived. So this probably requires more attention and discussion. So this is my last slide. I would like to summarize and give you some take-home messages. So what the work has shown is that there's strong attenuation potential for vinyl chloride and butyl acrylate in the East Palestine watershed. Natural attenuation should be considered when response actions are taken. And I don't think that has really gotten too much attention in the case of East Palestine, and probably similar accidents like this. The work also shows that molecular biological tools, qPCR, are a powerful tool for assessment and monitoring. But this can only be accomplished when you know biomarkers. Okay, you have to target specific biomarkers. Only then can you meaningfully and very effectively implement MBTs. Many of these biomarkers are known. For example, we know a lot about vinyl chloride degradation biomarkers. But not for all chemicals, for all contaminants, do we have complete understanding of these biomarkers. So what I'm trying to say here is that fundamental research on microbial degradation of these toxic chemicals that threaten environmental and human health must continue to develop a more complete suite of biomarker genes that microbial biological tools can then target. Not all of these biomarkers are known, and my fear is that there's a tendency to believe we've done a lot, maybe it's enough, and I'm saying it's not enough. We need to continue search for very important biomarkers so that we can do our microbial insights with the quant-array approach, can specifically target and quantify these biomarker genes in environmental systems. So we received NSF rapid funding. It's a fast mechanism to receive funding, but I argue it's not fast enough. The accident happened in February. We received funding in July. Our first sampling campaign was in June of 2023, several months after the accident. Based on the data that Gao Chen showed you, it appears that we did not capture the dynamic response close or immediately after the accident and the release of the contaminants. So what we are seeing could be after the bloom of these contaminant degrading organisms in the sediment, surface water, and we are seeing the tail end. We don't know. It would have been very, very beneficial if we had the opportunity to collect samples earlier. So that's why I'm saying, you know, NSF rapid funding is fast and great, but maybe we have to think about even faster mechanisms to allow people to do this type of assessment to get a more complete picture of what is going on, and also develop response scenarios for future accidents where we can handle things better. So we noticed that access to the site and access to information was not always easy to get. It was a challenge. And I can easily see major improvements. So samples were not easy to get because the site was closed off. We had to talk to local residents to get access to the creek. What was difficult? A lot of information that would have been very informative for us to design the experiments, the treatability study, were not readily available or became available after the fact. And there's still a lot of information that's not in the public domain. So there's also room for improvement. The accident response guidance is lacking. So think about an accident like this happening, and the people on the ground would have clear guidance documents, what to do if this happens, if that happens, and they can follow approved, and well thought out guidance documents. Maybe the response would have been a little bit different. For example, maybe the decision to have a controlled burn would not have been made. But these accident response guidance documents are not available. So I'm putting here ITRC, the Interstate Technology and Regulatory Council, with a question mark, because my suggestion would be this would be an appropriate, project for the ITRC to think about, develop these guidance documents and make them available. And hopefully in the future, when similar accidents likely will happen, we have a better and more thought out response, justified based on the facts on the ground. A last point I would like to make is community outreach and communication is a critical component of all of this. The residents of East Palestine suffered the most. And in our communication with the local community, we did not get the feeling that they felt the system has served them well. I think there's a lot of miscommunication, lack of communication, and maybe communication at a level that is completely inappropriate for local residents. I was shown tables of contaminant analyses that I, as a professional in this area, had a hard time understanding what was measured, how was it measured, what were the actual concentration, what does it mean? So it's, I believe, inappropriate to challenge local residents without background in this area to provide them with information they cannot digest. So there's a lot of room for improvement in this area of community outreach and communication. And maybe that's also a lesson learned, what needs to be done much better and differently in future accidents like this. With that, I would like to stop and give people a chance to ask questions. And we very much appreciate your time and attention to this important topic. Thank you very much.
All right, thank you so much, Frank, and thank you, everyone. I just want to note we do have a very large audience today, and there's a lot of questions that are being submitted, so not all of them will be able to be answered, but if you have further follow-up, I'm sure Frank would be happy for you to reach out to him or Gal, or you can also reach out to us at info@microbe.com, but we'll try to get through as many as we can. Frank, before we start, one of the questions is about using this information and how do we see that this data that we've generated is going to affect the EPA's plans to remediate the area. So can you talk a little bit about our thoughts on that, and then maybe some about the publication that we're putting together that could be helpful?
So to my knowledge, there has been very little talk about natural attenuation of these chemicals released. And the work that we have done points in a very clear direction. microbes exist in the East Palestine watershed that degrade vinyl chloride and butyl acrylate, and very likely also some of the other contaminants that have been released. So I cannot judge the relative contribution, of this process, but it's clearly a process that will have a significant impact on contaminant, fate, and transport. Detoxification is occurring at the site, and this is an important factor that needs to be considered in modeling and in interpretation of concentration measurements and everything. So the team here, Microbial Insights, and the team here in my group at the University of Tennessee, we have collected a lot more data than what we showed today. And we are putting together a publication that we hopefully submit to the journal Environmental Science and Technology in the near future, that summarizes all of these findings and highlights that in events like this, natural attenuation should be considered from the beginning and be part of the decision-making process, how to proceed with mitigating risk to the community. Did I answer this question?
Yes, that was very good. So as a follow-up on that, a couple of the people are asking questions about, one, how do we see Derailment in surface water, given that there is aerobic activity that's occurring there? So if you can talk about that, and then also why we don't always detect it, or we have very low concentrations in the qPCR data directly from the samples, but we're seeing more degradation happen in the microcosms.
Okay, so I've done a lot of work on Derailment in drinking water, in groundwater aquifers. And even when you have a homogeneous looking aquifer, sandy aquifer, you take a sample in one location, a core, and you take a core, less than a foot away. And the Derailment community will look different. In one location, it's high, in the other location, it's very low or not detectable. The point is, there's a lot of heterogeneity in the distribution of these organisms in an otherwise homogeneous looking aquifer. I showed you in the beginning this picture of the sediment where this connector stream is, and it was nearly dry. It rained overnight, and there was clearly water flowing the next day, a lot of water. We collected samples in the same location, maybe a couple meters away, and we collected multiple samples. And we found that Derailment detection can vary by orders of magnitude within a location. We sampled in June and in November, and we paid attention to go to exactly the same locations and sample those locations again. There's no way that you can go back to the same location and sample exactly the same spot again, because this is such a dynamic environment. My key point is, this is a very heterogeneous system. You cannot even consider that you do replicate sampling. There are no replicates. You have to just do enough sampling to get a general idea about the presence and abundance of these organisms, understanding that there's a lot of heterogeneity. It is not expected really to see Derailment in surface water because they are strict anaerobes. The surface water is most likely oxic, so they don't grow in the water. But if you have a bloom of Derailment in the sediment, and there's flow, quite a bit of flow in this connector stream, in Sulfur Run, in Leslie Run, you will stir up the sediment. So you release organisms that are growing in the sediment into the water column. This is what you can measure. Obviously, the abundances of these biomarker genes from these cells are low in surface water, but that is the explanation why you find them in surface water. But again, abundance is low, so a non-detect in surface water doesn't mean much, right? So what you would like to do you would like to collect sediment samples and enough of them to get some idea about the presence and distribution. Keeping in mind that work like this, you deal with a very heterogeneous and dynamic system, and you have to interpret the data accordingly.
That's a great answer, Frank. And I think it's important to point out too that there's never enough funding. One of the questions is about not collecting more samples, but there's never enough funding, whether it's privately funded or through grants, to really collect all the samples that we want. So like Frank says, we have to be judicious in where we choose the locations, and then interpret the data to the best of our ability. I don't know if there's anything else you want to add to that, Frank.
No, I mean, so, you can look at this funding issue, and we only have, everybody has 2 hands, but it's still a limitation. You should have 4. You can look at everything we've done. The glass is half full or the glass is half empty. With more funding, sure, we can fill up this glass a little bit more. But what we have done, all of the data we collected shows the interpretation of the data is pretty straightforward. And so I don't look at the limitations that we encountered as the glass is half empty. No, the glass is half full. What we have done provides clear information about natural attenuation potential, and this is the important information. If we would have sampled twice the amount of locations, sure, that would have been nice, but the overall conclusions would not have changed.
Perfect. Thanks, Frank. The other question, and there's a few people that have asked this, is about the interaction of having butyl acrylate there as well as vinyl chloride. So can you talk about, how the degradation of butyl acrylate could act as an electron donor for vinyl chloride, if that's a potential, or if any of the daughter products formed could?
Okay, so this is an interesting question, and I don't have a perfect answer because it depends certainly, the sequence of events when these chemicals were released. They were not all released at the same time, right? So one car was punctured immediately, the other one later. The behavior of these chemicals is a little different, but you can assume that some of these contaminants co-migrated into this watershed. And it's from what Gao demonstrated, butyl acrylate, and that's also true for some other of these other compounds that were released, they can be fermented or transformed to compounds that ultimately release electron donors that support reductive dechlorination, specifically hydrogen or formate. So yes, some of these chemicals released, like petroleum hydrocarbons, were also released, are electron donors for reductive processes that would also be expected to stimulate reductive dechlorination by dihalocucoides and dehalogenemonas. Gao talked about the observation that residents talked about, that their water now smells like rotten eggs, and that was never the case. It happened after the accident. So that basically is an example, right? Electron donor was released. It stimulated sulfate reduction in the aquifer and the sediment. Hydrogen sulfide was a product of sulfate reduction, and that chemical then made it into these drinking water wells and caused the smell that residents were reporting. So yes, many of these compounds that were released are electron donors for terminal electron accepting processes, including reductive dechlorination.
All right, thanks, Frank. There are 2 other things that people had a concern about. Is one, during this presentation, we really didn't get into other pathways that could be occurring at the site. And so that was one concern. So if you can talk about that, how we understand that there are other mechanisms that could be causing changes in the contaminants that were released. And then the second concern is that we're focused on this for train derailments when there are other modes of transportation for hazardous waste, such as trucks and things that can cause crashes.
Okay, so regarding the first question, other pathways, I think you mean other pathways of degradation or other pathways for fade and transport.
Yeah, it wasn't clear, but I would say probably for degradation.
Okay, so I mean, obviously, some of the chemicals like vinyl chloride that was released, right, it's volatile at room temperature or at ambient temperature. So a lot of the vinyl chloride would volatilize and be lost to the atmosphere. But it's very clear from the measurements, some of this material also penetrated the soil, went into the subsurface and into the creek water or the aquifer, and then was there and potentially a contaminant for drinking water wells. And I showed you that this didn't happen because EPA never detected vinyl chloride in any of these residential wells, suggesting that there was attenuation. We talked about microbial attenuation, but there are other processes that can act on vinyl chloride and possibly also butyl acrylate, abiotic processes that may be indirectly influenced by microbial activity. We did not assess that, but it is known that vinyl chloride can be abiotically degraded, but typically the reactive mineral phase that's involved in this abiotic process is generated by microbial activity. So we talk about microbially enhanced abiotic degradation. So when you think about abiotic processes, there's a process that generates the active reactive mineral phase, and that's typically microbial. So we have not specifically looked at this, but this probably also contributes to natural attenuation of these chemicals. Then there was a question about transportation. Whenever you transport, there's always a potential for accidents. It doesn't matter if it's on the road, if it's on the tracks, or if it's in the air. In the bigger picture, you could think about vinyl chloride production and using the vinyl chloride to make polyvinyl chloride in the same location. Then you don't need transportation of hazardous material, right? Then you generate vinyl chloride, the monomer, and polyvinyl chloride, the product, in the same location, not requiring transportation of hazardous material. So it's a matter of logistics, but I guess we are far away from that kind of scenario, but maybe in the future something to consider to avoid accidents like this.
Okay, Frank, there are quite a few questions about the testing for the dioxins and the PCDDs and the PCDFs that are being done at the site. I know our focus was on compounds that you have experience with and kind of limited in what we were looking at in the study. Do you have any knowledge that you could share about what's being done with these other compounds that were also spilled?
Well, so the other compounds spilled, I mentioned it, limited in amount of information. So it's still not clear, or it's very difficult to really know about how much of these different chemicals that were on these cars actually spilled and how much of the material was burned. It's not entirely clear. We chose to focus on vinyl chloride and butyl acrylate because based on the information we had at the time, those were the key contaminants, the key of greatest concern. Some of the other chemicals released were not as toxic or not toxic at all, or we knew that they were readily degradable. So that's the reason why we focused on vinyl chloride and butyl acrylate. Talking about the formation of combustion products that are of concern, like these polychlorinated dibenzodioxins and polychlorinated dibenzofuranes. So they were certainly formed. And I'm not sure if EPA has started measuring these chemicals in the region where they could come back down with rain, for example, on soil, agricultural soil. And the concentrations of these chemicals may be low, right? So detecting them may not be trivial. It's a complicated process. One concern that has been expressed is that these chemicals are recalcitrant and they bioaccumulate and biomagnify. So if if a fish has a low concentration or is exposed to a low concentration of these chemicals, they accumulate in the fatty tissue of the fish. Then the bigger fish comes along, eats it, and you see about a 10-fold increase in concentration. We call this biomagnification along the food chain. So it magnifies in concentration from one trophic level to the next trophic level. So basically in predatory animals or humans, eventually we could see biomagnification of these chemicals. So over time, you may see higher concentrations than what you would expect to see based on the measurements right now. So this is a concern, and EPA should carefully monitor these environmental systems, right, where we would expect to see the deposition of these chemicals after the burn, to make sure that people are aware and are notified should those concerns come true.
Okay, Frank, one last question and then we'll close out today. And if people have other questions, they can always reach out to us at info@microbe.com and we'll be glad to forward everything over to you and Gal for a response. But a couple of people have asked about, now that we've done this assessment, what is the plan moving forward? Are we going to continue to measure Derailment through the process? Are we going to use other tools like our biotrap samplers to look at the groundwater for collection? From our standpoint, what are we going to be doing next?
So that's a good question. I mean, the rapid funding is for 1 year. So, and it's a limited amount of money. We basically spent it on the activities that we have done. But, the chemicals we monitored, vinyl chloride and butyl acrylate, I don't believe pose a threat anymore in the environment, because they have been degraded or have been volatilized or they have been burned, right? So probably the concentrations are low now. The monitoring has to continue, that's clear. And monitoring the Derailment would certainly be another data point that would help to assess the long-term response and the long-term response of the microbiome to the contamination. It's an insurance, right? If you detect them, that you have a protective layer that these contaminants don't reach drinking water wells. So there's some value to it. But it's also very likely that all of those contaminants are no longer the real threat in this watershed. So what I'm mostly concerned about, what would have been very helpful for developing understanding to optimize the response for future accidents, was that we were not able to collect immediately after the accident. That would have generated a lot of very valuable information for us as environmental microbiologists and engineers to interpret, how quickly can a system respond to a perturbation? What can we expect in terms of natural attenuation from an environment that is suddenly exposed to a new toxic chemical? That would have been very informative, but we can't go back in time. So maybe this is a suggestion for a future accident, right? To make this rapid even faster. And I think that helps us all to be better prepared for the next accident like this and make decisions based on knowledge and scientific understanding.
Yeah, thank you, Frank. And as we close out today, just to be sure we're clear to everyone, is the research that we did was with the intention of trying to understand what is currently occurring in this system, and also to look for ways to improve, as Frank said, for the future, right? Hindsight's always 20/20, and we understand that when something happens, it's a stressful situation. So we're not, condemning anyone or how it was handled. We just we can't go back in time and change that at this point. So I think our goal in the research that we did was to understand better, and then to help with future planning initiatives, so that we have a better action plan and a better emergency response plan that can be applied across the board no matter what kind of accident occurs. And I don't know if there's anything you want to add to that, Frank, but I think it's important for people to understand that point.
Yeah, this is absolutely correct. I also, if you think about local residents, it's certainly encouraging and comforting for them that we can now explain natural attenuation mechanisms that detoxify chemicals that have been spilled and have been released. So you can't just tell them, oh, there's nothing in your drinking water, what many people have done without further explanation. So now we have a mechanism, a process, right, that can explain why nothing is in their drinking water. And I think this is very important also to communicate science to community members and tell them what academic research and all these companies are doing to help them deal with contamination and generate technologies involved in detoxification and cleaning up the environment. So all of this is important. And again, I think what has been done, you can change it, but there's room for improvement for the next accident. And that's also part of the effort that has been done and we are continuing to do under the grant we have from the National Science Foundation.
Well, thank you, Frank, and thank you, Gal, for taking time to do this presentation and inform the community. I value the interaction that we have with you guys, and I think the research that you do is really crucial to us having better response time, understanding pathways, and doing better management practices. So it's always an honor to work with you on projects.
Thank you very much, and we really appreciate the audience for listening to us. Thank you. And again, if you have questions, please, you can email me or Dora, and we try to answer your questions by email.
Yep. Yeah, thank you, everyone. Thank you, Frank. Thank you, Gal, for being here today. And we hope you'll join us next month on our webinar. Have a great day, everyone.
Bye-bye.
Bye.
Bye-bye.